A little bit of an introduction to this experiment. All living cells contain DNA. DNA is inside the nucleus, which is inside the cell of a living organism. So, every fruit, every animal, every human, every plant has cells. And every cell contains some sort of nucleus. The nucleus contains DNA, where we get our genetic material from. It's what we're made out of and what determines who and what we are. We are protected. The cell is protected from a nuclear membrane and enzymes called DNases, which can digest foreign or viral DNA upon invasions. Each cell in the human body contains 46 chromosomes. If you unraveled the DNA from each chromosomes and put the 46 segments end to end, each cell would contain about 2 meters of DNA. Each piece of DNA is around 4 to 5 centimeters long. Kiwi fruits have between 58 to 174 chromosomes. Speaking about kiwis, my task is to teach you how to extract DNA from a kiwi. Here are the ingredients that you will need. However, I will also explain and show you what to use throughout the process. This is a very entertaining experiment for me. I think every biologist, or at least every biology student, should try it. However, as we all know, there are precautions to take and rules to follow to ensure that you get the right test results and so that you're safe throughout the process. The first one, if you have long hair, it should be tied up just so that it doesn't distract your vision or no chemicals get on it. Second one, you should wear an apron just because it will ensure that no mixtures or chemicals will get on your clothes, because if they do, it will be hard to take off later. Third one, prepare the materials in advance just so that you don't have to run afterwards in between instructions. My suggestion is take specific measurement tools so that you don't add too much or too little and you have the right amount that is needed. But especially for this experiment, you should chill the ethanol 2 hours before you do the experiment. So it should be in the freezer for 2 hours, and it should be kept on ice during the experiment. The ethanol should be kept in the freezer for 2 hours before the experiment and on ice during it, because it precipitates the DNA and it does it better when it's cold. So here I am peeling the kiwi. The reason you have to peel the kiwi is because the outer skin is very tough and fibrous, and the skin acts as a barrier that would make it more difficult to access the cells within the fruit's flesh. So by peeling the kiwi, you ensure that the process targets the softer inner tissues, which contain the plant cells where the DNA is actually located. This makes it easier to break down the cell membranes and release the DNA during the extraction steps. Here, as you can see, I'm peeling it with my fingers. It did take me quite a few minutes. Then I realized that there was a knife around, so I took the knife, and here I am peeling it way faster. So by taking a knife, it would be more efficient that way. Here's the peel with kiwi. The next step is to cut it up into small pieces. I cut it up into 9 pieces approximately. Here they are. Then the next step is to squash all the kiwi in a little ball to make it into a liquid. The reason we mash the kiwi is to break open the plant cells, releasing their contents, including the DNA that we're trying to find. As you can see, it was quite hard for me to squash the kiwi because it's very hard still. So you can add 3 to 4 tablespoons of salt. It makes it easier since the salt breaks down proteins that are associated with DNA. Therefore, it's more easier. As you can notice, it did take some while before the kiwi actually turned into a liquid, most of it. So keep mashing it and squishing it. As you notice, I added another tablespoon of salt just because it was very hard. Here's the final product. The next step is to pour the liquid part into the test tube. However, as you can see, it was not very liquidy, so I took a funnel to pour it all in. Here I am pouring all the mixture into the funnel. It still didn't go in because there were big lumps in the kiwi mixture. If this happens to you, go and squash the kiwis again so that it's liquid like this. One reason it was very hard to pour the mixture into the test tube, specifically just the liquid, is because it had a lot of clumps from the kiwi itself. So I suggest cut out the white, really hard parts of the kiwi so that it's more easy to turn into a liquid. This is approximately amount of kiwi mixture that you'll need, and now we'll be adding hot water. It's important to note that I have added 1.5 tablespoons, or also known as 15 centimeters cubed, of washing up liquid to this mixture before adding hot water. The washing up liquid is known as dish soap or as the detergent in our experiment. The detergent helps disrupt the cell membranes and nuclear membranes, helping us expand the surface area and get to see the DNA better. Now, going back to the water, you should add 100 milliliters of it to the test tube. Note that I did add a bit too much of hot water into the test tube. Luckily, it did not affect my test results. However, it possibly could. So be careful and be really precise with the measurements and get specific tools which measure the quantities. After you have added the hot water into the mixture, you grab a stick rod and start to mix it. Mixing the mixture ensures that salt, detergent, and enzymes are evenly distributed, which helps break down the cell membranes and proteins. The next step is to add hot water, which is approximately 60 Celsius, into the beaker for 15 minutes. It is recommended to fill up the water until the mixture ends, so that all of it is covered. For future experiments, I suggest pour the hot water into the beaker first rather than swirling the mixture by itself. This way, the heat from the hot water will help degrade the enzymes faster, which are normally there to protect the nucleus from the invasion of foreign DNA. And while we're swirling in the hot water, it will give more enzymes a chance to be in contact with the hot water, making it faster to degrade. Here I am still mixing it. It will take a couple of minutes. Pay careful attention right now as I will swirl the mixture. It will change color, and it's because of the hot water, as I've said before, because it helps to break down cell membranes and degrade the enzymes, particularly DNases, which are in the nucleus to help protect the DNA itself. And as the membranes disintegrate and the enzymes become deactivated, the cellular contents, including pigments and other compounds, will be released, causing the mixture to change color. Once you have mixed it for a couple of minutes, leave it for 15 minutes. While waiting, you should fold some filter paper. Here are some options on how to do it if you are not sure. We use the filter paper to separate the clear solution containing the soluble components such as DNA while leaving behind the unwanted solids. If that was too challenging for you, here's option 2. And in the end, you should get this little cone that we add to the funnel to filter out our mixture. Here we finally have the complete mixture, a new test tube, and the filter paper in the funnel. I poured some of the mixture into the filter paper and now we just wait. I recommend pouring all of the mixture in at once so that more of it is faster filtered. If you feel like your mixture isn't filtering out, add some hot water into it and then pour it again into the funnel. It will take a while before it starts to filter out, but here you can see that a few drops have finally been put into the test tube. Here is a close-up of how the little liquid will look like. It's very clear. As you can see, the drops are pouring down. Slowly but steady. After quite a while, you will have about approximately this amount of liquid. That is enough. You pull away the funnel with the filter paper and you get the ethanol. Your test tube should be tilted 45 degrees and you slowly pour the well-chilled ethanol into the test tube. Pour about 10 centimeters cubed of ethanol into the test tube and you'll see a layer forming against the mixture. It's not quite clear, but here you will see the different layers forming on top of each other. The ethanol on top and the filtered kiwi mixture in the bottom. Now all you have to do is wait a couple of minutes and see how the DNA starts to float in the ethanol on top. Here is the final result. The little bubbles in the thread-like structure are the DNA. Do not feel discouraged if yours didn't turn out like that, or if you were not able to see the DNA, because my colleagues' work also didn't work out as it was planned to. Maybe try again and make sure to follow the right instructions with the right amount of measurements.